Here we're going to start to look at the function of DNA, which is really to express genes. And what a gene is it's a sequence of DNA that encodes a protein. So in these lectures, we're going to talk about how you move from a gene or a sequence of DNA to a protein. And the protein then is that sequence of amino acids. And the protein is going to be a unit, a functional unit that's going to actually have a job. And we've talked before about proteins and all the things that they can do for the cell. So proteins are going to do things like be transporters and be enzymes and be receptors, and they're going to do all of these jobs in a cell. So the cell has the ability to make various proteins based on the genes that it possesses. So what we're going to talk about in talking about how you move from a gene to a protein is these two steps. First, transcription. In transcription, we take the sequence of DNA and we transcribe just a unit, which is RNA. And so we're going to first talk about this transcription. Then the second phase is to take our unit of RNA and translate it to a sequence of amino acids. So this is called translation because here we're actually changing from nucleotides, which are RNA, to amino acids, which is a protein. And when we do this, we're going to be using basically a Rosetta Stone. It's the genetic code. And the genetic code is, it was worked out, so we had to work this out experimentally and discover how each set of three, which is considered a codon, a set of three nucleotides is going to correspond to a single amino acid. And so we'll be looking at this in detail, but just a little sneak peek down here. This is the genetic code, wherein you're seeing a series of three nucleotides encodes various amino acids. So here you can start to see some of those definitions written out. A gene is an organized unit of DNA sequence that encodes a functional unit. Now, one clarification I want to have here is, even though we typically talk about this as becoming a protein, and the protein being the functional unit. When that happens, the gene could be considered a structural gene. There are exceptions to this, or there are other variations of this, meaning that the functional unit that the gene encodes could also be RNA itself. And we're going to see that this RNA is actually going to be very useful within the cell. So down here we say sometimes that final product of a gene is just RNA. And the examples that we'll see throughout this lecture are the existence of something called ribosomal RNA. This is going to be important for making ribosomes. And then we'll also see transfer RNA. This is going to be important for that translation process. So when this happens and that final product is actually not a protein, but is instead just RNA itself, this is considered a non-structural gene. So even though when I typically talk about DNA moving through these stages of transcription and then translation to get to a protein, it's important to remember that this protein is not always going to be our only way to get to a functional unit. Sometimes the RNA step itself is going to give us useful things like ribosomal RNA or transfer RNA. And in that case, it's still a gene that's telling us how to make that RNA, but this is considered a non-structural gene. So this is just a little caveat before we move forward. Okay, so these processes also look a little different in prokaryotes compared to eukaryotes. In prokaryotes, remember, there's no nucleus. So this story is going to basically be the same, but we're going to take out the existence of the nucleus. So in prokaryotes, you have DNA. Step one is to take DNA and transcribe a unit of that DNA, and that what we transcribe from that DNA will be considered the mRNA. What the mRNA is it's just a little piece of the DNA. So the whole DNA is huge. And within this huge piece of DNA are going to be many different structural units, or things that could become structural units. Many different genes would be another way of saying that. So this strip of DNA here could be a gene, and this strip of DNA here could be a gene, and so could this little strip here be a gene. There could be many, and there are many genes within that long sequence of DNA. So step one is to take this piece of DNA that's going to correspond to a functional unit, or the gene, and transcribe the RNA from it. So really what we're doing in transcription is we're kind of copying down just the little gene sequence, and that becomes the RNA. The RNA then is going to be the thing that's translated. And this is because of course, we're not going to translate the entire DNA. We just want to translate the piece of DNA that corresponds to a functional unit, or the gene. And so we're going to first, step one is transcription. This is taking a piece of DNA and just transcribing our little piece of RNA. Step two then, once we have this little piece of RNA, step two would be translation. In step two, this is where we translate each series of 3 nucleotides into an amino acid. So that's considered translation because we're actually changing the language. We're moving from a nucleotide sequence to an amino acid sequence. Translation requires the existence of a ribosome. So in prokaryotes, this is all just floating around the cytosol. You've got the ribosome floating around, you got the DNA floating around, and then you've got the various players in this process that we'll see, which are going to be things like RNA polymerase floating around. Okay, so and we talked before about how this is possible for a prokaryote because it's relatively small and all of these players can find one another. The eukaryote is a little bit more organized. Of course, it has a nucleus, and we talked before about how it needs to be like this so that we can have organization. Otherwise, these players may not find each other. And my pictures here are not doing justice because the prokaryote is 10 times smaller, and that's something that we had talked about before. So the eukaryote is going to be a very large cell. And the eukaryote has its DNA inside the nucleus. And the rule is the DNA cannot leave the nucleus. The DNA is very protected, and we don't want to alter the DNA in any way. So the DNA always stays in the nucleus. The only thing that can come out of the nucleus then would be a piece of RNA. So this is why we need transcription. Transcription is this process of making a piece of RNA. The RNA is going to be a more mobile unit that can actually leave the nucleus. So the RNA also is not going to be the entire DNA, just like as we were talking about before with the prokaryotes. The RNA is going to be a little piece of the DNA that corresponds to a gene or something that's going to be useful and functional once we translate it. So we don't transcribe the entire DNA. We transcribe just a little piece of DNA that corresponds to a gene or a useful functional unit or something that will be translated into a functional unit. And so when we transcribe it, we make a copy of it, but it's in RNA nucleotides instead of DNA nucleotides, which are slightly different. The sugar in the sugar phosphate backbone is different for RNA. It's ribose instead of deoxyribose, which is what's found in DNA. So the nucleotides are different in that they all contain ribose instead of deoxyribose. And then the nitrogenous bases are slightly different because there is a uracil instead of a thymine. So there's a couple differences between the RNA and the DNA. But the biggest difference is the RNA is going to be single-stranded. And the second biggest difference is the RNA will be able to leave the nucleus. So the RNA, once it's made, leaves the nucleus, and then outside the nucleus is where it will find the ribosome. Outside the nucleus, the RNA will interact with the ribosome, and the ribosome does this process of translation. The ribosome actually uses the genetic code and will use a tRNA in order to bring in the amino acid that corresponds to each codon or unit of 3 nucleotides. So sometimes I like to think of this as a story. And the story that I like to tell is the DNA is like ancient textbooks. So they're these ancient books that are housed in a library. The library being the nucleus. And the rule is these ancient books are so special that they can't leave the nucleus. We want to keep them in the nucleus where they will be safe. So the DNA is in the nucleus, kind of like I've got this book in this library here. Now, if you want to use something from this book, what would you do? You would go into the library, and you can't take the book out. So what do you do instead? You maybe get out a little piece of paper and you copy down the parts of the book that you need. You write them down on a little piece of paper, and then that little piece of paper, of course, can leave the library. So you leave the library with your little transcript, the little transcript that you wrote down from the book on this little piece of paper. Now, this little piece of paper or this transcript is easily going to be lost. And that's going to be the same thing for the mRNA that leaves the nucleus. It's easily going to be degraded. It's not very stable. It probably won't stick around for very long. But as long as you have this little piece of paper, you can use it to, maybe it's a recipe or it's instructions, and you can use them in order to, I guess, make a protein or make whatever recipe was housed in that ancient library book. So the idea here is the mRNA can leave the nucleus, but it's not going to be very stable. But that's fine because it's going to be stable enough to interact with the ribosome and make this functional unit called a protein. Okay, so the purpose of this video is really just to focus on transcription, which is this first stage. This is where we're writing down the information from the DNA that we want to use. Or this is where we are making an RNA copy of the DNA sequence that corresponds to a usable unit, which could be translated to a protein or could be usable in and of itself as RNA if it's a non-structural gene. So our first step today is to look at, or in this video, is to look at transcription, which is the process of moving a double-stranded DNA to a single-stranded RNA. And an important part of transcription is we're not going to be affecting the DNA at all. It's going to be safe in the library. We're just transcribing. We're just writing down a copy of it. And that copy of it, we can go out of the nucleus with it, where we'll go and find a ribosome. So within the DNA, genes are organized as specific regions of the DNA. And a gene is going to be found between a promoter region of DNA and a terminator region of DNA. Between these two regions of the DNA will be the gene. So what this does is the promoter region of the DNA is going to be there along with, it's going to be near a regulatory sequence. And this is going to be where various proteins are going to be able to bind to the DNA, because these proteins are going to be the ones that do this process of transcription. So they kind of land on this promoter region of the DNA. That's kind of the docking zone. But it's got some very specific sequences that enable these things to dock with it. And these things that would dock with the promoter region are relevant for when we want to express this gene. So in general, the proteins that would be able to bind here are only going to be present within the cell or present within the nucleus during the time that the cell needs the sequences or the information housed in this gene. So the promoter is very important for when we're able to. But the first thing that happens is that proteins will dock and arrange themselves on this promoter region, and then we'll look at how transcription happens. So transcription is this process of copying down this usable sequence of DNA into an mRNA, or really an RNA code. And this RNA code is going to be complementary to the DNA. And then the gene will also have at the end of it a terminator sequence. So this tells these proteins that are moving along doing this process of transcription that this is the end of the gene. So the only part of the DNA that's going to be transcribed is going to be this usable region of the gene, or of the DNA. Now, of course, DNA is double-stranded, and so this means that it's got two complementary strands. This is the DNA. It's got both, and we had looked at this last week. It's double-stranded. It's got two strands that are complementary to one another. Either of them could correspond to the gene, but always one DNA strand, so one of these two, will be the template strand. And the other one technically is called a coding strand. That means that because it's complementary to the template strand, just like the RNA that we would be transcribing is going to be complementary to the template strand, and you'll be seeing that a little bit better as we move forward. The other strand is called the coding strand because it's actually going to have the same sequence of bases as the RNA that we're going to be transcribing from the template strand. Another rule, and this is going to be the same rule that we saw last week for DNA, is that RNA can only be built in the 5' to 3' direction, When we look at transcription, it's going to look a little bit similar to copying of a DNA sequence that we saw last week. The important thing to keep in mind is that we're using RNA nucleotides instead of DNA nucleotides. So once again, that's a slightly different sugar. Instead of deoxyribose, it'll be ribose. But because DNA is double-stranded, either of these strands could be the template strand versus the coding strand. So it could kind of go both ways. And we can kind of see some examples of that down here in this picture. So in this picture, we're seeing a great big piece of DNA. And housed in this great big piece of DNA, it looks like there's a gene A and a B and a C. And for gene A, the template strand is this bottom strand shown in the picture. That means that when we copy it, we're going to be, basically, when we copy this template strand, we're going to be copying something that is complementary to it, which will look just like the opposite strand, which is considered the coding strand. And so when we transcribe from this bottom template strand, we end up making DNA that looks just like the complementary sequence of DNA. We end up making RNA. I think I said that wrong. We end up making RNA that looks just like the complementary sequence of DNA. And over here for gene B, it looks like it's the same. The bottom strand shown here is considered the template strand. So the RNA that we transcribe from it ends up looking just like the other rail of DNA. For gene C, however, it's the opposite. For gene C, it looks like this is the template strand. And so the coding strand is the other strand, and that's going to be just what the mRNA looks like, or the RNA looks like. So either strand could be the coding strand or the template strand. It is going to depend on the position of the promoter and terminator. So if you look up here, for gene C, the promoter was here, so you start on this side and go this way. For gene B and A, though, the promoter was here, so you go this way for each of these, and you transcribe the gene that way. This tells us that a lot of genes can be housed within the DNA. Okay, so next let's try to get an idea of what is actually happening during transcription. So there's going to be 3 steps of transcription. Step 1, or stage 1, is initiation, or recognition stage. In this stage, various transcription factors, kind of a good name, so these are factors involved in transcription, and RNA polymerase are going to bind to that promoter region. So this is all of these factors kind of loading on to this promoter region, binding to it. So here's shown a transcription factor. It has bound to this promoter region of the DNA. And there could be various transcription factors. The transcription factors have to be present. And so this is what allows the cell to determine when it's actually going to transcribe a piece of DNA into RNA. We're not going to always be transcribing all of our genes. At any one time, only a subset of those genes are being used, and it depends on what transcription factors are there. So step 1 is you have to have these transcription factors, and they have to bind to the promoter region. Then RNA polymerase can bind. So it kind of uses the transcription factors to dock with the gene, or to dock with the DNA. RNA polymerase is kind of a good name because it's going to make polymers from RNA nucleotides. So RNA polymerase a little bit sounds like DNA polymerase because it's going to a little bit be doing the same job. Also in step 1, or this initiation phase, the RNA polymerase is going to create this open complex where it is breaking the hydrogen bonds between the 2 strands of DNA, opening up the DNA, and kind of revealing these nitrogenous base codes so that we have something that we can use in order to bring in our new nucleotides. This is very similar to what we saw with DNA replication. You have to pull the 2 strands apart so that you can begin using these nitrogenous bases to bring in and do some hydrogen bonding with what's going to be complementary. So we would be able to bring in the complementary nucleotides, and they would do their hydrogen bonding against these open or exposed nitrogenous bases. The open area will be about 10 to 15 base pairs open, so we'll be able to work with a 10 to 15 base pair region of DNA at a time. And the other thing that's going to be different between RNA transcription and then what we saw last week with DNA replication is this is only going to be happening in one direction. Because remember, there's only one template strand that we're interested in. We're not trying to do both strands of DNA. We're just going to use one strand of DNA as a template strand. And so the step 2 is going to be elongation. The RNA polymerase is going to move in one direction only. And as it moves, so where it opened up the DNA, those nitrogenous bases were exposed. And where they're exposed, those new RNA nucleotides are going to be able to come in and first form a hydrogen bond with the exposed nitrogenous bases. RNA polymerase then will move along the DNA. So it says that it releases the transcription factors and begins sliding along the DNA. As it slides along, it creates this open complex. So it kind of pulls the two strands of DNA apart, revealing those nitrogenous bases. It makes it so that those nitrogenous bases are exposed. The idea would be that as it moves along, there's this region of about 10 to 12 nucleotides that are exposed. And the exposed areas of nitrogenous bases are the ones where, now that they're exposed, these free-floating RNA nucleotides will be able to kind of come in and form those hydrogen bonds, stabilizing them near where they need to be. And then RNA polymerase will come along and polymerize these nucleotides into a chain of nucleotides, but this time it is RNA nucleotides. This is going to be called the RNA transcript. As RNA polymerase moves along, the area behind it actually reseals. So kind of an interesting thing that's happening here, and you have to look very closely at these pictures. This RNA transcript is only hydrogen bound to that template strand long enough for the RNA polymerase to kind of lock that sugar phosphate backbone together. Then the mRNA or the RNA transcript is no longer hydrogen bonded. It doesn't have to remain hydrogen bonded for very long. It's hydrogen bonded long enough for RNA polymerase to snap it together, and then RNA polymerase keeps moving along. The RNA kind of falls off from the DNA. The DNA reforms its hydrogen bonds, kind of closing up the DNA, and the little RNA transcripts kind of dangling out in the wind. So it uses the DNA as a template in order to figure out this order of nucleotides, but then they're released from the DNA because this mRNA transcript is going to be free and it's going to be movable, and really we want it to go on out of the nucleus and find a ribosome to interact with. And then we want that DNA to reseal behind it so that we're protecting and keeping intact the DNA sequence, because we want the DNA to be unchanged. So really we're just kind of unzipping it long enough to use that template or use that sequence of nucleotides to make our RNA transcript, and then kind of in the wake of RNA polymerase, the DNA reseals itself, protecting that nucleotide sequence, but allowing this mRNA sequence to exist. So this is the elongation stage, and this happens as RNA polymerase moves along, creating an open complex, but then allowing the DNA to reseal in its wake. Step 3 then is termination. This is going to be when RNA polymerase reaches that termination sequence of DNA. At this point, the RNA that has formed completely dissociates from the DNA. So there's our mRNA or our RNA transcript. It can now go off and go do whatever it's going to do next. We'll talk about that. And RNA polymerase can fall off because its job is done. The DNA reseals itself, and the DNA is completely intact. So now we have completed the steps of transcription. We now have our mRNA transcript, which is going to be complementary to the chain of DNA that it used as a template, and will actually be exactly the same as the opposite strand of DNA because they're both complementary to that template DNA. But the mRNA is different because now it's detached and it can go off, and we're going to talk about what happens to it next. And then very importantly, that DNA is completely intact because it just reseals, those hydrogen bonds reform between the two strands of DNA, and we have that sequence of DNA maintained, and it can remain safe inside the nucleus. Now there's going to be a couple things that have to happen to the RNA before it leaves the nucleus, and that's what we're going to talk about next. So in the 1970s, it was actually observed that, if we actually count up, all the possible genes that we could have in the DNA based on the amount of DNA. If we know that for every 3 nucleotides, it corresponds to 1 amino acid, that's going to be the genetic code. We're going to see that in more detail when we look at translation. But with this rule, every 3 nucleotides of DNA corresponds to 1 amino acid. Scientists in the 1970s kind of did some math on this, and they said, okay, well, if we have, let me just do some easy math here, if we have 300 nucleotides, then that should correspond to 100 amino acids. And when they did this math, really multiplied out times the hundreds of thousands of nucleotides, their math concluded that all of that RNA is not going to actually turn into a protein. There's more nucleotides than there ever will be amino acids. So this was a discrepancy, and it got sorted out when we realized that there are parts of the DNA that become RNA that are considered coding sequence, and there are other parts that are actually non-coding. These intervening non-coding regions are called introns. The useful coding regions are called exons because they're the expressed regions. And so something we have to talk about is RNA splicing. This is where these intervening regions are actually cut out and the exons are connected together. The introns for a long time have been considered junk DNA because they're not actually coded into a protein sequence. But now we're beginning to understand that these little introns may actually have some functions in the cell beyond just being junk DNA. Though I do believe a lot of the literature will still call them junk DNA. We haven't completely figured out what all these intervening sequences are good for. But here's what scientists figured out. They said so the gene is considered everything between the promoter and the terminator, and so we get an RNA from that. The intervening sequence are called introns, and these parts are going to be removed such that the introns can then be fused together, and that becomes what's called mature mRNA once you have the introns removed. And so these junk DNAs actually are degraded, or maybe we don't completely understand what happens to them. But we do know that they are removed, and they're not going to ever leave the nucleus and go and interact with the ribosome to become a protein, or to be translated into a protein. So splicing of the exons together and removal of the introns is one of 3 things that has to happen to what's called pre-mRNA in order to have what's called mature mRNA. It's only the mature mRNA that actually leaves the nucleus and goes out to the cytosol to interact with the ribosome and be translated into a protein or an amino acid sequence. So we're going to look at these 3 things that have to happen to the RNA after it is transcribed. The 3 things can be summed up as capped, spliced, and tailed. So the cap is called a 5' cap. This is where at the 5' end of the RNA sequence, a modified guanine is added. This modified guanine is added during transcription. It's added about when the RNA is 25 nukes long. And the cap is important because it can be recognized by cap binding proteins, which help to pull it out of the nucleus. Studies have been done and shown that if it doesn't have this cap, the RNA rarely leaves the nucleus. The cap also prevents degradation of the mRNA out in the cytosol. The RNA is not very stable, but the cap helps its stability and helps it get to the mRNA, helps it get out of the nucleus, remain stable, and then get to the ribosomes where it can be translated. I've mentioned splicing. That's where the introns are cut out and the exons are spliced together. There is something called alternative splicing, where there's actually more than one way to splice an mRNA. And so you could actually cut out this exon as well and splice exon 1 directly to exon 3. And we'll look at this more carefully in lecture this week. The third thing that has to happen is it has to be tailed. So at the 3' end of the RNA, a poly A tail is added enzymatically. This is just a long sequence of A nucleotides to create this poly A tail. The poly A tail is important for getting it out of the nucleus and preventing